Hello, and welcome to demola. And today's video we are covering plant cells. So we're going to go through all of the organelles, comparing them to animal cells, and maybe even chuck in a cheeky bit about beta glucose. No time to waste. Let's get straight into it. Okay, so first of all, we're going to talk about the organelles inside of plant cells. So, know that eukaryotic cells, right? DNA is arranged as linear chromosomes, membrane-bound organelles. Please try and remember differences between eukaryotic cells and prokaryotic cells because, again, this is something that stays with you all throughout the course. Okay. But let's actually look at the plant cells specifically. So this is a plant cell. And I've drawn a couple of the important organelles and I've just also annotated the ones that are specific to plant cells. So you can see here, different to the animal cell, we actually do have a cell wall. So I'll actually highlight them along the way as well so you know what we're talking about. So this is the cell wall, this outer bit here. Yeah? Made up of cellulose, very, very important to know. They love saying that in the exams. And we'll talk more about cellulose later. What else you've got here? You've got your pits. So pits are kind of like this narrowing or the invagination of the cell wall. And the reason why that's important is just because it allows transport of molecules. Similar idea when we talk about plasmodesmata as well, but these are now gaps. So again, it allows communication or transport of materials between cells, but these are the gaps instead. Now we've got a permanent vacuole, which contains cell sap. It helps with the cell's integrity, and its membrane is called a tonoplast. We've also got the amyloplast, which, to be honest, doesn't really get mentioned too much, but essentially it's starch-containing granule. So it stores starch. Yeah, it contains starch granules. And then we've also got chloroplast, which is the main guy. Chloroplast is going to be a site of photosynthesis. There's other organelles that we haven't mentioned because they are in animal cells as well. But here you've got your Golgi apparatus, the smooth endoplasmic reticulum, mitochondrion, and then also you've got rough endoplasmic reticulum here. And obviously dotted around, you've got a key player, ribosomes as well, ATS. So those are the main organelles that we want to focus on. And obviously I haven't mentioned this as well. Got our nucleus and our nucleolus here. So those are organelles inside of your plant cells that are good to know. And we've defined them as well. But we're going to look about some of them in a bit more detail. So cell wall, its main function is structural support and a barrier. Yeah, and like we said, it's made up of cellulose, which is a polymer of glucose. We're going to talk about it later. What's also important, it's mentioned in the spec. I don't think it comes up too often, but obviously these things are good to know. Middle lamellae. So it's a thin layer, mostly containing of this substance called pectin, right? And it holds the cell together, and it's between adjacent plant cells. So if we just do a diagram, say that's cell wall 1, that's cell wall 2, this pectin is in the middle, and it sticks them together. Yep. We've also got our chloroplasts. So you'll focus more on this in topic 5. So that's why I haven't done too much information here. But in terms of the chloroplasts, just good things to know for now is that this kind of cytoplasm mimic is called stroma, right? Then these interesting structures here, specifically the ones that I've drawn as like these hash lines, are called thylakoids, right? And a stack of thylakoids is something that we call, so a stack of them is called a granum. Again, we'll talk about more in topic 5. And finally, lamellae. Again, same word. So sorry about it. But lamellae join your thylakoids together. Yeah. And the main thing about chloroplasts is they contain the pigment chlorophyll, which is going to be very, very important for photosynthesis. But again, that's topic 5's problem. Something else as well that you should be able to do as well as differences between animal and plant cells. So similarities are going to be everything that you can say for eukaryotic cells. Those will stay the same. However, differences are going to be cell wall. Right, so plants have the cellulose, animal cells don't have the cell wall. They only have the cell membrane. Permanent vacuole versus temporary vacuole. So I think it's very common for people to say that animal cells don't have a vacuole. They do, or some of them do, but it's normally temporary instead of this big permanent one. Also, in terms of an organelle that they contain that the other doesn't, plant cells have chloroplasts. Animal cells have centrioles, but plant cells do not. So that's something a lot of people miss as well. Plant cells do not have centrioles. And then also, just energy storage molecule. For plant cells, it's starch in your amyloplasts. And in animal cells, it's actually just glycogen. So again, good things to know in terms of differences to say in an exam. Okay, now to be honest, because I studied SNAB and the videos are mainly for SNAB, we don't actually talk about microscopy often on this course. However, just because we are doing this for like AQA and OCR, so I feel it was good to just add in a tiny bit. So electron microscopes, they use beams of electrons, whilst your other alternative, the light microscope, uses rays of light. So that's the difference. In terms of the resolution, which is like how clearly you can see things separate from each other, the electron microscope is a lot better. So electron microscope is 0.2
nanometers. And just in relation to a meter, nanometer is times 10 to the minus 9. So times 10 to the minus 9. Whereas a micrometer is times 10 to the minus 6. So you can see quite a significant difference in terms of the resolution between nanometers, which is for the electron microscope, and micrometers, which is for the light microscope. So that means you can see these smaller organelles. So your ribosomes, your endoplasmic reticulum, they're quite smaller. If you see that in an image, it means it was from an electron microscope, because a light microscope wouldn't pick it up. However, with an electron microscope, because you're shining the beam of electrons, they have to be dead. So you can't view live specimens with an electron microscope, which is the most major disadvantage. There are different types of electron microscopes as well, so transmission and scanning. So with your transmission electron microscope, that gives a 2D image of internal structures. So you cut a really, really, really thin slice, you shine the electrons through it, and you get that 2D image, right, in a lot of detail. However, the scanning electron microscope is a bit different. This time, you get a 3D image of the external surface, yeah? So 3D image external surface, and that will give a lower resolution than transmission electron microscope. So, in the context of A-level biology, typically, if you see it in a question, it will come from a transmission electron microscope. So if that's a 1 marker, transmission electron microscope is probably your best bet. And also, it's very important to be able to see different organelles. So I've got a plant cell here, and I'm just going to highlight different structures here. So you can see here, this massive thing here is your vacuole. Yeah? So you can see it's a different color because of the cell sap, and it doesn't have any of like your ribosomes or anything. So a different color cell sap, and you can see the membrane of it is called the tonoplast. So that's one thing. Another thing you have here is your chloroplasts. So probably a bit easier to see the chloroplasts when they're focused in on, but you can kind of see the granum or the stacks of thylakoids that we talked about earlier here. And you can also see that this is quite a large organelle. So especially if you know it's a plant cell, just be thinking what's logically going to be here, chloroplasts. Same thing if you had muscle cell, will logically be a lot of mitochondria. So you've got that. What else have you got here? You've got your nucleus. So nucleus you can spot because it's got that double membrane, and you can see here that massive black dot in the middle is going to be your nucleolus. So that's another thing that you can notice. Remember about what you know should be there and try and relate it to the image. And finally, another thing that we've got here is the mitochondria. So you can't see it in too much detail. You'd have to focus in. And to be honest, that's a topic 6 question when we go into respiration. But just know that it is there. And I've also just got examples here where I focus in on some things. So it's really, really good to be able to describe what you see and then say why it's this organelle. So first of here, I see a stack of membranes. I'll just try and highlight it to make it clear. I see a stack of membranes, and I also see these vesicles surrounding it. Yeah, or these dots surrounding it is found to say an exam. So that means probably we're dealing with the Golgi apparatus. Yeah. Similar thing here. What am I looking at? I can see a thick membrane. Okay. I can see this. This is your granum or your stack of thylakoids, and this is the lamella in between. So I'm just describing what I see. That must be a chloroplast. Yeah. Again, here, what do I see? Well, you haven't done this one yet, but you would say I see the foldings or the inner membrane, and then you can also see just a bit of molecules around it. So this one, again, we're not going into too much detail because it's not yet, but this is a mitochondrium. And finally, again, you're seeing stacks of membranes. You're seeing stacks of membranes. You're also seeing, although maybe it's a bit harder to see, can you see ribosomes? Potentially, ribosomes on it. So because of that, this is probably rough in the plasmid reticulum that you're seeing. So just kind of make a note of these things. And finally, what I also want to see is this one, hopefully you can make out as a cell wall. You can see you have both sides of it. So that's a cell wall, and you've got the cellulose in the middle. And something that I'm hoping you can appreciate as well, do it in green, is this here. So that's a gap in the cell wall. So that's your plasmodesmata. So again, I'm just showing you things, but you need to really be able to practice in your own time about taking an electron microscope image and seeing what's going on. Okay, and last thing in this introduction to plant cells, we need to talk about starch and cellulose. So, starch is the energy storage molecule in plants, whereas in animal cells, you're using glycogen. Now, starch is actually made up of 2 different polysaccharides. So, amylose and amylopectin are what you're going to use to make starch. And you can see the percentage composition there. Amylose 20%, amylopectin 80%. Now, just because starch isn't our main focus this video, we're going to kind of go a bit quicker through this so we can spend more time with the other one. But you can see here, amylose is a straight chain. So it's made up of alpha 1,4 glycosidic bonds, whereas amylopectin is branched. So it has the 1,4 and the 1,6. And you can see I've done my best to draw a molecule there. So this would be amylopectin. You'd have a bunch of these in a starch molecule. So you can see highly branched, a loss of glucose. It's not easily soluble, so it resists osmotic pressure. These are things that make it a very good energy storage molecule, starch. And also because of the branching, it's quite compact as well. So starch, great. Cellulose is a bit of a different one. So cellulose is used to strengthen the cell wall. Yeah, it's a polysaccharide of beta glucose, which is an important point. So alpha glucose, beta glucose, different things. You can see here in alpha glucose, if we look at, and we'll just label them here, carbon 1 and carbon 4, you can see the OH groups are both down. Yeah. Now, if we use beta glucose, you can see on carbon 1 it's up and carbon 4 it's down. So those are the main differences. Apologies, I didn't complete my diagram. This should be like that. And then this should also be like that. OH. My apologies. I don't like missing things out. so that's glucose, yeah? Now, why is this an important point? Well, first of all, different molecules. Second of all, it affects the bonding. So cellulose is another straight chain, but you're going to form beta 1 for glycosidic bonds, right? Because it's beta glucose. Now, if you have this like this, you can see here the groups on opposite sides, the hydroxide groups on opposite sides. They are not close enough to react. Yeah, that's what I've put written here. They're not close enough to react. So what you need to do is inversion. So you can see here. Can you see how, if you just look at this oxygen here, can you see how the molecule is inverting the whole way in the cellulose chain? So we're inverting it so that the OH groups can be on the same side. And if the OH groups are on the same side, then that means that we're forming very, 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 very nice straight chain. That's the important thing to know. So it's really irked me that I haven't put the extra carbon there. Those are all 4 carbons. 5 carbons, so. But don't worry, cool. Another thing that's important to know is you can form cellulose microfibrils. So cellulose microfibrils, they happen like this. So I've got this image from Save My Exams, but you can see here, we have our hydroxyl groups, right? And you know hydroxyl groups can form hydrogen bonds. Why? The oxygen and the hydrogen, very, very polar bond. So there's a dipole in there. There's a delta negative with the oxygen, delta positive with the hydrogen. That should still make sense to you if you're a biologist. If it's a chemistry, it should be bread and butter, right? Now, they can form the hydrogen bonds because there's an attraction between them. So like we said, this one is delta plus, this one is delta minus. There's an attraction, we can form a hydrogen bond. And that's going to happen to make your cellulose microfibril. So joining of cellulose chains together forms a microfibril, which will go on to make a fiber, so on and so on. We'll talk about that a bit more in future lessons, but that's a kind of overview into cellulose and its importance. Okay, I feel like I've done a lot of talking for 3 slides, so I'm going to give myself a bit of voice rest. So we've got 4 questions really for you to try here. Pause the video, have a go, and then we'll go back with the answers. With the last one, I think that's only 2 marks. Okay, so here are your answers now. You have to use your knowledge of cellulose and compare it to what you see in front of you. So we'll get the highlights out and we'll go through it. Now, I can see straight off the bat with chitin, I can see that nitrogen group. I can see here that we actually do have inversion of the chains. So chains are inverting, that's good. And we can see that you have glycosidic bonds as well. So these are all things that I'd definitely be thinking. I can mention this when it comes to exam. Yeah. So look here. Inversion, glycosidic bonds, they love it. Yeah. Now, I saw the nitrogen group. So I would have got 3 marks for this question. What I probably would have checked a bit closer to exam is the carbon. So you can see here we've got obviously 1, 2, 3, 4, 5, 6, 7, and 8. So 8 carbons. So this isn't a hexo sugar. This is 8. So make sure you're looking at things like that when you're answering questions like this, and you'll be able to maximize the marks. Now, middle lamellae, again, in terms of the content of the question, I think like the role of middle lamellae, it doesn't, mitosis has nothing to do with this. I feel like they've added in mitosis here just to throw people off. So I just explain the role of middle lamellae, which you can do, yeah? Between adjacent cells, or maybe new cells because of mitosis, so you're adding a bit of context, and then it holds the cell walls together. That's the important bit. And then finally, when you look at the electron microscope here, we've seen an example like this. So we should be happy to say that this that I've circled is a plasmodesmata, yeah? And if it's plasmodesmata, we know that that has a function to make a connection between the 2 cells, or cytoplasmic connection, maybe to add a bit more depth to your answer. And then that allows the transport communication between the cells. So those are good things to know. Okay, let's wrap this up with the A-star tips. So what is good? Properties of eukaryotic cells comes up again and again. Remember, with SNAB, especially synoptic thinking. So you're thinking, I need to know this. Yeah, because it's come up again now. Being able to describe the functions and features of organelles, not only in plant cells, but in animal cells as well. I think it's just a very important skill to be able to see what's going on, to describe what's going on, and then also to say the function. And typically, if you get an electron microscope image, that is what they will want you to do. And that leads on to the next one, electron microscope. Know what you're expecting to see for each organelle, and then apply it when it comes to exam. They typically don't need you to be too specific about what you're seeing, as long as you're describing it well. Then, formation of the cellulose change in microfibrils is important because it's going to link onto like a core practical. It's going to link onto the next topic as well, and about sustainability and everything like that. So, very important to know how that's made because they can ask you so many questions about it. Now, what's not good to do? Differences between animal and plant cells. I think probably not too common of a mistake, but definitely possible. So make sure as well things like knowing the centriole is only in the animal cell is really, really nice and it's going to get you marks. Knowing that animal cells is temporary vacuoles as well, going to get you marks. So make sure you're doing that. The features of the organelles can be difficult to describe, but I'd say look at mark schemes. Maybe even ask AI, ChatGPT, and it should help you. So like, for example, with Golgi, you're looking for a stack of membranes that's getting increasingly longer, and you're also looking for these vesicles surrounding it. Yeah, make sure that you know that's what you're looking for Golgi. If you see it, say it. Then also polysaccharides with the structure and function also can be confused in terms of just knowing starch specifically plant cells, glycogen specifically animal cells, starch, glycogen, energy storage, cellulose, structural support. So just know differences like that as well. But that is it for this video today. So I hope it's been really, really helpful. If it was, please like and subscribe to the channel. We're getting very, 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 very hot on 1K subscribers, which would be amazing to achieve. Any questions at all about anything I said about plant cells, about A-level biology, heck, even A-level chemistry, put them in the comments below and I'll do my best to answer in a timely fashion. We've also got our TikTok and our Instagram, both at demola. Links will be in the description below. But that's all for me today. Thank you so much for listening, and I hope it's really, really helped. Bye for now.